So first I wanted to thank the organizers for allowing me to give this talk. So I'm a computational neuroscientist working in the Chen lab and the Bushaki lab close with the experimentalist, and my main goal is to understand how collective activity in the hippocampus gives rise to memory and navigation. So I would like to start by pointing out that there is this prevailing idea in neuroscience that all neurons are equal and that they contribute equally to the neural code. But we know that that's actually not the case. So neurons are not equal. This is a little bit related to the talk we heard yesterday about the constraints on learning, but there are several constraints on neuron activity. So for instance, if we look at the fighting rates show heavy tail distributions in which a minority of neurons does the majority of the spiking. But this is not just related to spiking, but also neuronal correlations are asymmetrically distributed, but also the number of the encoding properties. So for instance, the number of place cells a given cell has also show this log normal distribution or this type of heavy tail distributions. And also the morphology, so even the axon diameter or like the spine size shows this type of heavy tail distributions. So the question is, how are population patterns actually affected by these constraints? And what I'm interested in this particular talk is how contextually flexible communication is achieved in the hippocampus, considering that the underlying units will be asymmetric and will have different type of contributions, and how these patterns support memory consolidation. So we performed these two set of experiments. On the first set of experiments, we recorded the hippocampus with this synapse probe, which allow us to record from all major hippocampal subfields. So to give an idea of how information flows in the hippocampus, so it enters through the dentate gyrus, then it goes to the CA3, CA2, and the CA1 regions, and then it projects outward to the neocortex. So with these synapse probes, we can get single unit data across all these single areas at the same time in awake animals. And also we perform a second set of experiments using neuropixels, which allowed us to record from CA3, CA1, and also the retrosplenial cortex, and the retrosplenial cortex being an indirect projection from the hippocampus, which is an association area that contains a spatial map as well. So for all the analysis that I will show, I only employed putative classified pyramidal cells. And so the problem is, okay, so how do we understand communication between the different populations? So building on the works, previous works in the field, we use canonical correlation, and I will explain a little bit why I like canonical correlation for this. Basically to measure low-dimensional subspaces that maximize the correlation between two different areas, what people often call communication subspaces. But since we have several areas recorded at the same time, we can actually check a pairwise interaction while accounting for the activity of the remaining areas using the partial canonical correlation. And one of the things that I like about the canonical correlation is that it's fairly simple, but it's also fairly interpretable. So for instance, for a given neuron in the CA1 region, and since I have multiple areas recorded at once, I can check, okay, what's the contribution to the CA1, CA3 subspace, for instance, and what's the contribution of that same neuron to the CA1 retrosplenial subspace. And the contribution will be given by the weights of the given cell for a given subspace dimension. So that's the first thing we started. We started by looking at the subspace weights across different subspaces and check how they were correlated or not. So in the right set of plots, you will see the synapse probe. So this is comparing the CA1, the absolute weights for a given cell across different subspaces. And for the right plots, you see the neuropixel recordings. And what you can see is that the neuron weights are highly correlated across different subspaces, meaning that if a neuron communicates in one subspace, it's likely that it's also communicating through another subspace as well. And we find this highly correlated activity. So at this point, it was a little bit paradoxical when I started seeing these results, because I mean, if we take these results at face value, it would imply that the identity of the inputs and outputs are somewhat mixed in CA1, and that to us seemed a little bit suboptimal to be mixing the identity of the inputs and outputs. But then a different type of solution can appear if you think that maybe the identity is preserved in the subspaces themselves, and then the subspaces are rotated, for instance, one with respect to another. And this rotation is what preserves the identity, for instance, of the input to the output transformation. So how do we measure the subspace rotation? So to measure this, we use principal angles. So we measure the principal angles between the different subspaces. So we started by measuring with the synapse probe within the CA1 region. So we basically compared how the subspace, the rotation, what happens between two different subspaces, for instance, CA1, CA3 versus CA1 retrosplenial, and we compare these to the same subspace. So for instance, CA1, CA3, but just computed in different time intervals. And what we found is that in general, the different subspaces show larger rotation compared to the same subspace. And we could take this one step farther with the neuropixel recordings and actually measure for each individual area, for instance, C1, C3, and retrosplenial, if there is some areas that show larger rotations compared to the other ones. And we found that CA1 showed significantly larger rotations compared to these other two areas. And we sort of capture this idea with a little bit of, with this schematic, in which you can see if you are bounded to use the same set of neurons, because that's the constraint you have, your neural constraint, you need to use these 4 neurons. So for instance, in this type of case, you can have the input subspace. The first dimension will feature neuron 1 and neuron 2, and the second dimension will feature neuron 3 and neuron 4. But for the output subspace, you get that there is this recombination that takes place, and you will see that neuron 1 and neuron 3 now belong to the first dimension in the output, and the neuron 2 and neuron 4 for the second one, and that's a way in which you can preserve the identity despite needing to use exactly the same neurons. So then we tested whether, okay, this type of combinatorial principle can be applied in order to communicate in context-dependent manner and achieve like flexible communication. So we performed these experiments with the neuropixel recordings in which the mice run on a linear track, on a physical linear track, and then for a given set of trials, and then we switch the mazes and the surrounding context in a very short interval of time, and the mice run in a different novel environment for the same set of trials or a similar set of trials. And importantly, this period in between was short, relatively short, so we can actually measure the exact same 3 neuronal populations in these 2 types of contexts. So the first thing we did in this case is to measure that the total amount of information that was being transferred in these 2 mazes was more or less remained constant. So for this, we actually approximated mutual information linearly through the canonical correlations. And when we compared, we saw that roughly the total amount of information that was being transferred remained more or less the same. Then we look at the memberships, or we look at the subspace weights of individual neurons, and we saw that the neurons the weights were highly correlated between the 2 different mazes in these 3 different regions. But then the interesting thing happened when we actually took the subspaces from one maze and projected it to the second maze in order to understand and see how much of the correlation it actually explained the second maze. And what we found is that for different mazes, the correlation that it was able to explain dropped basically to chance levels. So it was not distinguishable to just employing like multi-unit activity. And our control for this was longer novel maze sessions in which we could divide the trials into the first set of trials, the second set of trials. So if the maze remains constant, then actually the correlations generalize between the first half and the second half. So we see that this type of combinatorial principle sort of also applies into communicating in a context-dependent manner. So at this point, I want to make a little parenthesis and ask the following question. So what's actually, what makes some neurons fire more? What makes some neurons communicate across different environments? So what we did here is to basically divide the neurons recorded into subspace members and non-members just based on their absolute weights during pre-maze non-REM sleep. So that was before even the animals face the maze that would define them as members or non-members. And we saw that in this pre-maze non-REM sleep, the future members already fired at faster rates. And remember that these are all pyramidal cells. And actually, there were also bias in their anatomical location, in particular for the CA1 and the retrosplenial region. And they also have different encoding properties within the maze sessions themselves. So for instance, it's well known that the hippocampus, so if you take the population activity and you do like UMAP analysis, for instance, in order to find the manifolds, low-dimensional manifolds, you see that the full population captures both the position of the animal and the trial structure, which you can see here in the rightmost plot. So then we did the same dimensionality reduction, but just employing subspace members or subspace non-members. So this would be like around 1/3, 1/4 of the whole recorded population. We found that just employing members more or less captured more or less all the information that was present in the full population, while only employing non-members performed much worse. So out of this parenthesis, we now wanted to ask the following questions. So how these subspaces actually support learning, or how are they related to learning? So to try to tackle this problem, we did the following. So we had these noble maze sessions, and they were preceded by sleep periods before and after the noble maze session. So what we look was whether the subspaces computed during the maze, whether they will be reactivated during the post-maze session, indicated that these subspaces had been learned. And what we found is that, especially during non-REM sleep and during ripples, the subspaces, both CA1, CA3 subspace and CA1 retrosplenial subspace, were significantly reactivated comparing the post-maze with the pre-maze sleep. So since we know that during the ripple periods, there is spike in activity, and this spike in activity, as we saw in the previous talk, it recapitulates more or less the type of behavioral patterns that the animal did during the maze, which is called replay events. We wanted to see whether this subspace communication somewhat biases or contributes to the replay events. So what I'm showing here, it's 4 different examples. The top plots, you can see the CE1 LFP, which shows the defined ripple oscillation, this oscillation that takes place in a very short time interval. In the middle, you can see the spike in activity of all the recorded cells that spiked for those set of trials. And the heat maps below show the posterior probability of the positions. So basically a Bayesian decoder that was trained during the maze runs. Then we assess how the activity during the post-maze sleep, these post-maze periods, recapitulates the activity that was seen during the maze runs. And what we found, and we sort of expected this, is when the CA1, CA3 communication was high, we saw that there was these periods in which there was a replay of a behavioral trajectory. But what we didn't actually expect to find is that when the CA1 retrosplenial subspace, the correlations increase, we didn't see a change in replay. But we actually saw that when the CA1 retrosplenial communication increased, replay tended to decrease. And we quantified this both at the level of pulling all spiking events together and also at the level of the single sessions, and this was fairly consistent. So since we saw that there was this pattern in which there was this differential reactivation pattern related to these 2 subspaces, we wanted to understand, okay, is this related to the information being consolidated in terms of like single pair interactions? So we measure the spike transmission probability between pairs of neurons by means of the cross-correlograms. So the cross-correlograms should show a defined peak within 5 millisecond window, suggesting that there is information being transferred in that window. So we computed this for the different subspace members and non-members. And what we found is that for CA3 members to CA1 members, there was an increase in spike transmission probability during post-maze sleep compared to pre-maze sleep. That's fine. But what we didn't expect to find was that, again, CA1 members to retrosplenial members, they didn't show any change pre to post-maze sleep. But on the other hand, we did find that the CA1 members to the retrosplenial non-members, they show a decrease in spike transmission probability from pre-maze to post-maze sleep. And we believe this suggests that there may be simultaneously different type of plasticity rules being applied in the same sleep session. So to get some ideas and conclusions, so we found that there is a minority of non-random neurons that communicate across subspaces and context. And these neurons are actually, what I'm saying that they are not random, they are deep cells in the pyramidal CA1 layer. They are layer 5 neurons in the retrosplenial cortex, and they are also likely to be athorny cells in CA3 region. And we believe this also has implications on future recordings and even maybe BMIs. So if you can target these specific subpopulations, then you can get more information with the same number of units recorded. We also find this type of combinatorial coactivity principle that sort of explains the context-dependent or the input and output identities. And this type of rotation principle has already been shown and worked extensively in the motor system, for instance, comparing preparatory activity to motor execution. And we believe this type of rotation or combinatorial principle may be a general principle for brain function, especially when you have these constraints in which you need to use the same number of cells or the same type of cells to do several different type of tasks. And we showed that the subspaces support what we believe may be different plasticity rules that are simultaneously co-occurring. And there is this debate, especially in hippocampal physiology, whether ripples promote LTP or ripples promote LTD, meaning long-term potentiation or long-term depression. And what we actually find is that both of these things can be simultaneously co-occurring. And with this, I want to acknowledge some people. First of most, Mishi, which performed all the experiments in the Bushaki lab, and also Seish and Yuri for all their valuable feedback, and to you for listening. Thanks.
Oh.
Great talk. So hippocampal activity, especially like in naturalistic environments, has the potential to be pretty sparse and high dimensional. So I'm wondering if you have thoughts about this kind of analysis and like the dimensionality of the subspaces that you would need to use and how that would scale with the complexity of the environment.
Okay, so yeah, actually, the subspaces themselves were not that much high dimensional, actually. So you could more or less if you had like 100 neurons recorded in each region, you would like get maybe 5-6 dimensions that already captured, that were significant according to surrogate testing. But I think we're capturing a little bit of this high dimensional type of idea with the fact that the same neurons can be recruited in different coactivity sort of modes. And which is, I think, something different, for instance, what we saw yesterday in the cortex, in which, because, I mean, according to what we discussed with Emily yesterday, this may be like off-manifold type of perturbation. So maybe this high-dimensional hippocampal pattern is seen in the fact that the same neurons can be recruited in different type of ensembles according to the task demands. And I do expect that the complexity of the task should increase the dimensionality, but unfortunately we just have this linear type of mazes in which you record several areas at once in order to get this idea.
In the retrosplenial cortex, I was curious which layers are you recording from? Is it all layer 5 pyramidal cells? Because layer 2, 3... I've studied this with Yuri before. I just am curious, I
think we are covering actually all because I mean the type of penetration we are making with the neuropixel, I think it targets both layer 2, 3 and also layer 5.
So you had distance, and one of the plots is like 0 from, for the superficial cells.
Yeah.
For the hippocampus, it's very clear, right? There's a million papers on that. But in the RSC, it's a little more complicated. Like people like Ahmed Ammar have found these really weird cells in layer 2, 3 that are supposed to have to do with head direction tuning. And then a lot of the cells in RSC are tuned to like mixed modalities, like vision and movement or sound and movement. And so do you think like sub-select it? Like is it just all the cells in RSC?
Yeah.
Or did you sub-select layer 5 and layer 2, 3 or?
No, they are all cells, and that actually the layer definition, the layer separation, that's where we got the best results out of the. So if you see the purple curve there, it's significantly deeper compared to the green curve. That's where we got, and I think we are more or less getting this definition, this distinction between these layer 5 neurons being the ones that encode this space and are communicating with hippocampus, and layer 2, 3 neurons doing a little bit something different. Because I mean, we sort of find like this deep motif between C3A thorny cells that should be a little bit deeper. I think we are not completely targeting them in the penetration, but then deep C1 cells and also layer 5. We have some ideas or some like crazy ideas about how development might have shaped this type of things. But
is that, if you think of the functional or behavioral correlates of the pyramidal cells in C1 and C3, right, what you're looking at for those is just simply place tuning, and you assume that the cells are basically tuned invariantly to anything but place, right?
Yep.
Almost explicitly or exclusively place. And of course,
Can you switch seat mode, please?
what? Oh, yeah. Turned it off by accident. It's not surprising that you would see replay when the two areas that generate replay on
we
their
expected
own
that.
together. But it's surprising to me that you don't see replay during the C1 RSC communication. And I'm wondering, I guess my question really is that if you looked at different populations of neurons in RSC, either anatomically or in terms of their multisensory tuning, I bet you would see replay. Because the RSC is doing so much, and it's not like C3 to C1. Like RSC, that many inputs. RSC is getting so many more inputs. I think depending on the task is very important, but even just with this data, I think looking at this physiology of the cell types, because if you're looking at excitatory cells and you look at layer 2, 3, if you use the same classifier, they don't look like pyramidal cells in C1. I'm just curious. For somehow I expect under certain conditions, basically you would see the replay, and I'm just curious how far you've gotten into that so far.
Yeah, totally.
I love it.
We actually expected that also that replay would actually take place. So a little note on replay here is that, we are using mouse, and the mouse place fields are not as nice as the rat place fields. So we are actually, what I'm doing is pulling all together all the recorded cells in order to construct the Bayesian decoders.
Yeah.
Even interneurons, everybody, just the only threshold I'm using is the spatial information that they have. So when you see the replay, you see replay across the whole pool of recorded cells across all three areas. And that improves quite a lot the type of posterior probabilities that we are getting. So I don't know exactly what we would get if we subdivide further the retrosplenial into different types of populations. But I definitely can check the tunings of the retrosplenial cells to see if there is something that may be worth digging a little bit further. Thanks.
Okay, we'll have to leave it there. Thank you, Joaquin.